Let's learn about recombined DNA technology in this video. In the nucleus, we have the chromosome which is made up of the DNA molecule, and this DNA consists of the nitrogenous base pairs. So, we will be learning about how we can cut these nitrogenous base pairs, or in simple words, a sequence of these base pairs, and insert it into another chromosome. Let's begin by taking an example of insulin preparation by using this recombined DNA technology. So we are aware that a human DNA is made up of gene, that is, it is a sequence of nitrogenous bases. Now we identify that in an entire DNA molecule, let's consider this is a fragment of the gene which is coding for insulin. So we will cut that particular fragment from the human DNA, and we will insert this fragment into a bacterial DNA, and once we insert this particular bacterial DNA will keep on replicating. Once this will replicate, we have large quantity of insulin which will be produced. So this is a diagrammatic representation of it, like this is an insulin gene, this will be our plasmid. Now what is plasmid? We will learn about this in the next video, but just for basic understanding, keep in mind that plasmid is a circular and extra chromosomal DNA, which means that let's consider this bacteria. So this bacteria is having its own chromosome, and this particular chromosome is controlling all its bacterial existence, while this plasmid is extra chromosomal, which means even if we make any changes here, it won't affect the survival of the bacterium. Therefore, we take this circular chromosome or circular DNA only, which is called as plasmid. So once we join this, we insert this back into the bacteria, and finally the bacteria will keep on replicating, because of which it will make large copies of insulin, and we are having insulin available at large quantity. Now host is an organism which will carry this particular recombined DNA. In this case, the bacterium is the host, while this particular circular portion will be called as the vector. So in simple words, a vector is actually a DNA molecule which will carry our DNA, that is our gene of interest. Now let's look at the word called as recombined. From this example we get to know that recombining involves combining two different DNA molecules, and now we will learn how can we break this particular fragment, how to join it, and how to insert this. The history of rDNA technology. So the scientists Boyer and Cohen recombined two plasmids called as PSC101 and PSC102. Once these two were combined, they were added in the E. coli. So E. coli is acting as a host. Now the next question is, why only these two plasmids were used? It was seen that this PSC101 plasmid had a gene, and that gene made the plasmid resistant to tetracycline, which means that if tetracycline is added, the bacteria, for example E. coli, won't die. Its survival chance will increase because it is having a gene which is resistant to tetracycline. Similarly, this plasmid had a gene which is resistant to kanamycin. Now from this experiment, what do we understand? That if we recombined these two and add into the E. coli, the resultant E. coli will be having resistance to two antibiotics, that is tetracycline as well as kanamycin. This is the first experiment of this rDNA technology. Now let us learn how do we cut the gene of interest from this entire DNA molecule. For this example, let us consider we want the insulin gene which is present in the entire or large human DNA. On this human DNA, there are recognition sequences, which is a palindrome. A palindrome means a sequence which reads same in the forward and backward direction. For example, let's consider this is a DNA. It is having two strands. One is 5' to 3' and the second anti-parallel to it is 3' to 5'. Now, here we have GAATTC in this particular direction, while in the exact opposite direction we have GAATTC. So the sequence is same in the forward and backward direction. Such palindromes are present in the human DNA and they are easy to recognize, and this is the point where the enzymes will act. So in this diagram we have a DNA and these are the enzymes which are acting on it. So they break this particular DNA or they separate this DNA in such a way that we have TTAA here, and similarly TTAA would be present on the strand, that is upper strand and lower strand. So this part is free, even this part is free. Here we can add our gene of interest, or we can see that this is how the rDNA could be combined. In simple words, let's consider this particular portion was our human DNA. So we have separated this. Now this particular portion which is joining is our plasmid DNA. For joining it needs something, right? So this would be provided by the sticky ends, and these sticky ends means the free nucleotides which are present. And how do we get them? By cutting it at the recognition sequence. To cut this DNA fragment, we need the enzyme which is called as restriction endonuclease or molecular scissors. These are the examples of the enzymes. Let's consider the first one, which is ECOR1. It is obtained from E. coli, and this is the palindrome sequence on which the ECOR1 will be acting. So the sequence is always fixed. This ECOR1 will act here in one strand and here in the lower strand, because of which we have this sticky end which will be formed. So we have two sticky ends which are formed. The nomenclature of this enzyme involves first alphabet, which is always capital, and it will represent the genus. The next two alphabet represent the species. The third alphabet is again capital and represents the strain, and the fourth alphabet is the Roman number, which is 1. It represents the order of discovery. Let's take another example, which is TAC1. So T represents the thermus, which is the genus, and it is always capital. The next two, AQ is the aquaticus and 1 is the order of discovery. So this is a DNA fragment where ECOR1 will be acting, because of which there is separation of this DNA strand and we have this sticky end which is available. Now this is called as the staggered end, that is some portion here is not having nucleotides, this is free so that other nucleotides can add up. This type of cleavage is the staggered end cleavage. Hint 3, restriction endonuclease will fragment this DNA into exact two halves. This type of cleavage is called as blunt end cleavage, as the end which is formed is blunt. There are no free nitrogenous bases available in the fragmented DNA. The next question arises is, how do we choose these restriction endonuclease while designing an experiment? For that, we usually are aware of the gene sequence. For example, let's consider we are interested in inserting the insulin gene. So we know what is the human DNA sequence, we know the gene sequence of insulin. Along with this, we are also having knowledge that what is the nitrogenous bases present around this particular insulin. So once we know that, we can exactly use those particular molecular scissors and get our insulin gene cut or separated from this human DNA. Let's consider our gene of interest, that is this DNA, is not having sticky ends. It thus becomes difficult to add such DNA into the vector DNA. We therefore have to add this sticky ends externally. So these linkers are chemically synthesized DNA molecules. Since they are chemically synthesized, we are aware of the base sequence, that is what nitrogenous bases are present here. Usually they are chosen in such a way that the restriction endonuclease can easily act on them, they can be separated, and we have this sticky end added to the foreign DNA. Now this foreign DNA is ready to get inserted into the vector DNA. This is linker. While if the attachment of this DNA molecule is having preformed sticky end, then this will be called as adapters. So the only difference between both of them is, while adding this chemically synthesized DNA molecule, if the sticky end is present on it, they are called as adapters. If the sticky end is not present, and again there is additional step where a restriction endonuclease will be used to break this or to cut this, then it is called as a linker. Once we have this vector plasmid ready, that is, it is having the sticky ends here, there is a phosphate group present as well as an OH group. Because it is a DNA molecule, so the OH group is always present on the 3' end and the 5' end will be having a phosphate. It is necessary to remove this particular phosphate group to prevent the recircularization of the plasmid, that is the dimer will be formed, because of which we use the enzyme called as alkaline phosphatase. This enzyme removes the phosphate group and there will be the OH group which will be added, and once phosphate is removed, the DNA ligase cannot act, so it will prevent the recircularization of the plasmid. Once our foreign DNA is ready and we want to attach or insert this into the plasmid DNA, we have to join these so that it will turn out to be one entire unit. For this joining, the enzyme called as DNA ligase is used. This enzyme is also useful to join the linkers or adapters to the foreign DNA. In simple words, wherever we want any nitrogenous bases to be joined to each other, we use this DNA ligase enzyme. It will form a covalent bond between the two DNA fragments. Let's consider this is a nitrogenous base. It is having a deoxyribose sugar, and there is a second nitrogenous base which is present here. The fifth carbon is having a phosphate group attached to it, while the third carbon is having an OH group attached. So the DNA ligase will ensure that a phosphodiester bond is formed between these two groups, and this DNA ligase always needs the NAD plus for its functioning. Nuclease enzyme. So the role of this enzyme is exactly opposite to that of DNA ligase. Where DNA ligase involves in forming the phosphodiester bond, this nuclease will involve in breaking the phosphodiester bond. So there are two types, exo as well as endonuclease. Now this exonuclease means it will act on the terminal side or outside of the DNA fragment, and it will cut these fragments, that is the phosphodiester bond will be broken, while the endonuclease will act on the phosphodiester bond which is present in between the DNA. So we have a gap which will be formed. The example of exonuclease involves nuclease Bal31, and the example of endonuclease is nuclease S1 and DNase 1. So here we come to an end of part 1 of the recombined DNA technology. In the next video, we will learn about different examples of the host and the vector which are used in the rDNA technology. Thank you.